Welcome everyone. Good morning or good afternoon to you. And my name is Marie Latulippe, and I'm the Director of Science Programs at IAFNS, or the Institute for the Advancement of Food and Nutrition Sciences. And we're excited today to continue our series highlighting the work of the USDA-ARS Human Nutrition Research Centers. And we've actually already covered Tufts, Arkansas, and Beltsville. So today we're covering the Western HNRC, and all of these programs that we've previously completed are available on our website if you'd like to check those out. So as we start, just a bit on IAFNS. We are a nonprofit organization focused on advancing food nutrition science for public benefit. And the way we work is we involve scientists from 3 different sectors: government, industry, and academia, to support collaboration and to catalyze knowledge. And at IAFNS, we do fund research through an open and competitive process. And actually, all of our funded projects are viewable on our website if you want to take a look. We also conduct our work by leveraging pooled funding, which is combined with in-kind contributions from both the public and the private sectors. And hopefully, when you registered, for those of you who are interested, you noted that we are an approved provider of continuing education credits by the Commission on Dietetic Registration. So we'll provide access to that certificate shortly after today's webinar. And in terms of how we'll run today's program, hopefully you see a control panel like this, or one that looks like this on your desktop. And what we'll do is we'll go through all of our speakers today, and we'll use the remaining time for questions and discussion. But as we go along, please type your questions in for any of the speakers that intrigue you. We will keep the introductions relatively short to allow more time to hear from our panelists. And we do also have a bios document on the website where you registered. So if you'd like more detail, please go there. And I'm very pleased to start by introducing our first speaker, Dr. Charles Stevenson. And Dr. Stevenson is a research leader at the USDA WHNRC, and he's also an adjunct professor of nutrition at UC Davis. And his ongoing work at the center is examining the associations of typical diet with the intestinal microbiota and the potential impact of microbiota on systemic immune activation and inflammation. So we'll turn it over to Dr. Stevenson, who will provide us with a quick overview of the center and its work. So Dr. Stevenson.
Hi, good morning, everybody, or good afternoon, good evening, as appropriate. I hope you can see my slides. I just have a brief introduction to the Western Human Nutrition Research Center. We're one of 6, as we just heard, human nutrition research centers. Beltsville, Grand Forks, Tufts, the Children's Nutrition Center at Baylor, as well as the Children and Infants Nutrition Center in Arkansas, and then our Western Human Nutrition Research Center. So we're located on the campus of the University of California, Davis. We're USDA scientists, but we're really a hybrid center, and so we have a lot of UC Davis students and staff in the building. Our mission within the USDA mission, the mission for our center is stated right here. It's fairly broad: to improve dietary, really to improve the scientific basis of dietary guidance coming from the US government. We do a lot of team science. Our facility is built around doing collaborative human nutrition studies. Human nutrition studies are inherently collaborative. This is a picture of one of our groups that finished up our USDA nutritional phenotyping study in 2019. And you'll hear about team science from Danielle LeMay and Mary Cable in just a few minutes. I'm just going to give you a brief overview of the center. We are on the Davis campus. We've been here since 1999. Before that, we were located at the Presidio of San Francisco. So I'm the center director as well as a research leader. We have 2 research units that consists of Let me just get my pointer. 13 research scientists. So Brian Bennett is the research leader of the Obesity and Metabolism Unit. His scientists are listed here. And then I'm also research leader of the Immunity and Disease Prevention Research Unit. So since we do collaborative human studies, in addition to our 13 research scientists who each have their own labs and individual research foci, we have 2 core support units, the Human Studies Unit and the support labs, Physiology and Bioanalytical Support Lab. So we have about 90-ish people in the building when we counted last month. A little less than half of those are USDA scientists and staff. Remaining are students and UC Davis staff and other visitors. So the 2 research units have different foci. So immunity and disease prevention, we really focus on how diet, sometimes specific nutrients, affects the immune system. So for example, Ryan Snodgrass and I, we focus on direct dietary effects, sometimes specific nutrients like vitamin D or fatty acids on the immune system, as represented here. You'll hear from Danielle LeMay and Mary Cable. A major focus of our unit going back, I guess, 7-ish years now, is how diet affects the immune system and other measures of health through the microbiome. And the 5th member of our team in the IDP unit is Bess Caswell. Her specialty is dietary intake analysis. In the obesity and metabolism unit, there are 6 scientists. Kevin Leggero focuses on stress biology. John Newman is an analytical chemist, focuses on lipids, is also a member of the West Coast Metabolomics Center. Brian Bennett, genetics and nutrition in animal models as well as humans. Lindsay Allen, our past center director and also a recent ASN president, is a center scientist and working on nutritional content and quality of human milk. Liping Wang is focusing on zinc biology, and Nancy Keim, her work focuses around dietary intervention studies. So just very briefly, as with the other human nutrition centers, we recruit study participants, not so much via telephone as the slide's indicating here anymore, but more web-based. We have provide dietary interventions, which are often food, of course, meals, sometimes dietary components like dietary fiber in a current study. And most of our studies in recent years are what we call free living, where study participants are not residing in the building. We do have a 12-bed metabolic unit, and we do have historically have done a lot of residential studies, and in the near future that's going to be a focus again, particularly from Brian Bennett and collaborators' work on the NIH-funded Nutrition for Precision Health study. And then, of course, we collect biological specimens and other test data. So very briefly, the metabolic kitchen looked like this late yesterday afternoon, fairly quiet. However, often it looks like this. We have staff preparing food, either to provide to participants in our dining room, but this slide is really showing preparation of food to be packed out and taken home. And a lot of the food is stored frozen. Of course, some of it's always fresh, but in order to feed a lot of people, we need to take advantage of our multiple walk-in freezers. So yesterday, one of our studies testing a variant of the dietary guidelines for Americans, 2 different recipes, 2 different meal plans. Study staff were packing up food in these what look like carry-on airline bags to take home for participants in a particular study. So that's a common paradigm at the WHNRC. And then just very briefly, we do have core support labs to focus on physiology assessment, pulmonary function, metabolic rate, DEXA for body composition, bone mineral density, cardiopulmonary fitness, and so on. And our bioanalytical support lab collects all those samples, urine, stool, blood, takes them upstairs to the support lab, processes samples specifically for each particular study, aliquots, stores in the freezer, and retrieves them for later analysis, either in the bioanalytical support lab or in research scientist labs. So that's the end of my introduction. And speaking of research scientists, now you'll hear from Danielle LeMay and Mary Cable about their specific research program.
Wonderful. Thank you so much, Dr. Stevenson. And we have Mary Cable joining us. By way of brief introduction, Dr. Cable is a research microbiologist at the Western HNRC, and the Cable Lab is currently investigating how foods rich in complex carbohydrates and polyphenols can influence the structure of the adult gut microbial community and colonization resistance to pathogenic microbes. So we'll turn the platform over to Dr. Cable to learn more about this work.
Thank you so much, and I'm really excited about the opportunity to speak today, so thank you for the invitation. I'll just begin with my disclosures. I get funding from the National Honey Board. I'm co-PI on some of Danielle LeMay's grants from the California Dairy Research Foundation. I also am on some grants with Brian Bennett from NIFA, and then of course USDA employs me. So now for my talk. I have one more disclosure, which is the first part of this talk, some of you may have seen before because it's one of the major studies that sort of launched a lot of the work that I do in my lab. So stick with me until sort of midway and the end, and you'll hear some new stuff. So I wanted to just start, since the theme of this is describing the work that we do here at the Western Human Nutrition Research Center, with the primary focus of my lab, which I structured around using 16S rRNA sequencing to identify potential microbe-diet relationships, generate hypotheses, and then test those relationship hypotheses using in vitro fermentation methods. The nutritional phenotyping study that Charles mentioned has been a huge source of lots of human stool and lots of human metadata to go along with that stool, which is a microbiologist or microbial ecologist's dream come true. And Charles Stevenson was the PI of that study. There's a design and implementation paper out, and then there have since been a lot of follow-up publications using data from that study, and that's the data I'll be presenting today. Our objective looking at data from that cross-sectional study was to identify robust diet-gut microbiome relationships in a diverse, healthy US cohort, correcting for age, sex, and BMI. So the key differences from some of the other cohorts that were out at the time are that they weren't necessarily diverse. They didn't necessarily exclude individuals based on chronic disease or taking medications, or they weren't necessarily big enough to be able to adjust for age, sex, and BMI. So that's what we wanted to add to the literature with this data. And we did it using 2 to 3 automated self-administered 24-hour recalls from these healthy individuals. and comparing it to the gut microbiome data that was collected from a single stool sample that the subject collected at home and it was frozen here at the WHNRC within about 24 hours. There were a few, I think maybe 10 or so, where that wasn't the case, but for it mostly wasn't an issue. We used Zymobiomics DNA extraction kit and then sort of standard 16S sequencing, and we used samples for which we got at least 10,000 sequences per sample. And then as any of you who are familiar with human studies and or microbiome data will really clearly understand is that there's a huge amount of variation that's not necessarily well characterized. So we started with exploratory analysis, and I found that sort of on a first pass, the bacterial taxa varied significantly by self-reported ethnicity. And we worried that maybe ethnicity, self-reported ethnicity, was acting as a summary variable for individual variation in diet pattern and or maybe combining it with variation in genetics. So, and we did do a follow-up analysis that showed that the types of foods consumed tended to vary by self-identified ethnicity as well, which sort of reinforced this concern. And so since we wanted to understand macronutrient microbe relationships, we didn't want them to be obscured by captured diversity and needed to recover relevant details about that variation. So as many of you are probably very familiar with, Abigail Johnson had published this paper with this amazing new method of structuring FNDDS, so the dietary data, into a tree format, which mirrors the way that we structure taxonomy for the microbes, and made the statistics much easier to compare between microbiomes and dietary patterns. So the way that that works, in case you're not familiar, and I kind of like to have it re-explained to myself many times as well, is that in the FNDDS, food is categorized into 9 major categories. So for example, milk and milk products, eggs, legumes, meats, and then within those 9 major categories, there are 5 levels of specificity. So it is a great analogy to how we classify bacterial taxa. This is an example within milk and milk products of the higher levels of specificity, and how, if you have an example of the products that are reported by subjects, how those end up breaking out into a tree. So you have these second level milk and milk drinks, cream and cream substitutes, milk, dessert, sauces and gravies, and cheeses. And you can see how all of these different types of things that are milk products could potentially have very different impacts on the gut microbiome that we might not capture in the things that are already within the databases. So structuring out this variability could help us a lot. And then Dr. Johnson also went through and used UNIFRAC, which is a tree-based distance metric to analyze the differences in the food patterns. So this is an example of how the UNIFRAC metric works. It was originally published by Catherine Luzapone a really long time ago, and is really frequently utilized now. And again, I just think it's really helpful to kind of describe how it works. This tree here sort of represents what might be all of the foods reported in a dietary study, like the one we did in the cross-sectional study. And then if you wanted to compare diet patterns between subject triangle and subject circle, for example, all of the foods from subject triangle would be here, and all of the foods from subject circle would be here. And the gray bars are showing foods that were differently, I believe the gray bars are foods that are different between the two, and the black bars are foods that are the same. And so the distance metric will be the total number of lengths between the foods divided by the foods that are different. So you'll have a higher number if, I might have said that wrong. Forgive me, I'm nervous. But anyway, it's a fraction, and it's a division of the distances between the foods. So Dr. Elizabeth Chin took our dietary data and cleaned it up together with some dietitians here at the center and Yasmin Bouzine in Dr. LeMay's lab, so that we could have this type of data for ourselves as well. And when we took that and compared it to the gut microbiome data from the subjects who had consumed those foods, we saw that first, if we applied the whole food dry weight to the data on the tree, there was a good correlation between the diversity of the food and the phylogenetic diversity of the gut microbes. That relationship, you can see the R value increased if we applied the total grams of carbohydrate per food to the tree. However, if we used total grams of protein or total grams of fat, that was not the case. The number went down. And if we use total grams of fiber applied to the tree, we got our strongest correlation yet. So we went through and used k-means clustering to structure that data, the dietary data, into patterns of consumption based on just clusters. So the k-means clusters are shown here for fibers consumed from individual foods. And we can see that diets that were high in fiber from fruit excluding berries, which in our study is primarily apples, bananas, avocados, so fruits that tend to be really high in pectin, those individuals also had a lot of the pectinolytic bacterial genera Lactospira. I wanted to see if that could also be detected with data that was already available. And we did see that there was almost a linear relationship between the HEI whole fruit score and Lactospira as well. This was the only significant relationship between the whole fruit score. However, when we went back to total carbohydrate, the pattern of total carbohydrate consumption had a strong association with bifidobacterium, which is a great, generally there's a hot topic, around bifidobacterium. And so we were really excited to see what was in this cluster. And it was mainly things that you would not expect to be associated with a healthy diet. So grain, pasta, French fries, and carbonated soft drinks. And then when we went and used HEI scores for grains or whole grains or refined grains, there was no linear relationship with bifidobacteria, and instead it pulled out a non-linear relationship with a fermenter, which suggests that this particular refined grain score is picking up some other variable that's sort of incompletely correlated with refined grain. So the summary of this is that we feel pretty confident that Lactospira is associated with consumption of fiber from whole fruit, probably pectin. We don't know what's going on with bifidobacterium and French fries, and we definitely need more characterization of carbohydrate structure within our dietary databases. And I have a feeling Danielle's going to cover some of that information in her talk a little later. So the interesting thing about our carbohydrate observation versus our fiber observation is that defining fiber and measuring it is a little bit tricky. For nutrition labeling purposes, the FDA has defined fiber as non-digestible carbohydrates that are 3 or more monomeric units, as well as lignin. And these are intrinsic and intact in plants, but can be isolated or synthetic in our foods. And then this key feature I think is interesting. They have to be determined by the FDA to have physiological effects that are beneficial to human health. So that's a pretty high bar to clear. But they do have a huge list of fibers that have now been defined as fiber. So when we look at the total carbohydrate variable, total grams of carbohydrate that we applied to our diversity of diet tree, what are we actually looking at? We're looking at the total grams of the food minus the sum of the measured fat and protein. So we're actually capturing a lot of probably unmeasured variability between foods. Whereas for total fiber, we're capturing a direct measurement after a specific digestion process. And one concern I had when I looked at this is that certain things that we might consider fibers would actually not make it through this process for measuring fiber. And perhaps the fibers that we're picking up in that total carbohydrate variable, where there's an association with bifidobacterium, could be among those that wouldn't be in that fiber variable that's measured. And so resistant starch type 3 is one of those fibers. It's created by gelatinizing starch at elevated temperatures, slow cooling, which results in recrystallization and then sort of cycling through that. And the reformed crystals can be A-type or B-type as measured by X-ray diffraction. And all of these different aspects of how those fibers crystallize can affect whether they are resistant or not to amylase digestion, that would affect whether or not they're measured in and identified as fiber. And this is sort of a summary from a great review about resistant starch type 3 of the different things that can affect that. So what do we do now with this 16S information, which we're getting used to seeing and being really hand wavy about, because it's it basically gives us more questions. We don't know exactly what fibers are in total carbohydrate, and we think that when you eat fruit, you get a lot of lactospira. So is that good? Does it always happen? Is it because of the fiber, or is it because of some other aspect of the food? So now we have all these great fun questions to answer, and they're much more difficult and time consuming to answer than just doing a 16S analysis. So Dave Storms, who's the project manager in my lab, set up this really great continuous flow fermentation system that has 4 parallel fermenters that run in tandem. And during the pandemic, when there weren't many people allowed in the lab, Dave came in and figured out what media to use and ran these pilot studies to see how long it takes us to get sort of a stable community inside our fermenters. And so now we have this great system where we don't have to just take a snapshot of the microbial community under specific growth conditions. We can stabilize that community with a media that's kind of been published a number of times, and we tweaked it a bit ourselves. And then spike in things that we expect that community to have a reaction to and see what happens over time. So that is what we have done. And we are first looking at 4 different fibers. This data is really still in the works. And so I'm blanking a little bit out here because I don't want to be really held to it. It's brand new data. But we're testing 4 different fibers. One of those is pectin. And this is stool from 2 individuals that were in the FO100, the phenotyping study that I showed earlier. One of them we know consumed habitually a lot of fiber, and the other we know consumed habitually not a lot of fiber. However, you can see that when we take their stool sample and spike in pectin, they both have the same bloom of lacnospira over time, no matter how much fiber they normally consume. And this is only an N of 2. We have a lot of other fermentations now conducted, but not necessarily analyzed. And hopefully this data will hit the presses by next year sometime. But we're really excited about this and the ability to test these correlations that we're seeing in the data. With that, I want to just switch gears quickly because I did promise I would show you new stuff. From the same cohort, Dr. Elizabeth Chen, again, who did a lot of amazing work while she was here, looked at the amount of lactose intake in these folks. And I think a lot of us have had the question, can lactose act as a fiber for certain individuals? If you're lactose intolerant, you're not processing that disaccharide. It's only got 2 monomers, so it's not technically a fiber by FDA definitions, but you're not processing that disaccharide in your small intestine, and so it's making its way to the large intestine. And previous studies have shown that gut microbiota may be capable of adapting to dairy consumption in lactose intolerant or lactase non-persisting individuals who don't express the LCT gene. However, dairy foods like aged cheeses and yogurt contain very little lactose, and so studies that have used dairy consumption as sort of a proxy for lactose intake are probably overestimating lactose intake. To add a level of difficulty, the ASA24 does not output lactose, but the NCC Food and Nutrient Database does. And so Dr. Chin, in order to estimate lactose from ASA24 data, mapped ASA24 foods to the NCC foods and estimated the recent lactose intake for our subjects in the phenotyping study. And she found that lactose intake varied and lactase persistence varied by ethnicity and by genotype, or phenotype in this case. She also looked at genotype and it varies by that too. But here we're showing that lactose consumption is significantly lower in lactose non-persisting individuals, so those who might be intolerant. And that difference is more significant than when you look at dairy consumption. When we looked at the interaction between lactase persister status and the amount of habitual lactose intake or recent lactose intake, we saw that when we looked at the full cohort, which might actually be including a lot of variability that we aren't really hoping to capture, that the family Lactobacilliaceae was increased in individuals who were non-persisters but who consumed lactose. When we looked at a Caucasian Hispanic only subset, which decreased the amount of variation in sort of diet pattern plus genotype and avoided skew. So for example, none of our Asian participants were lactase persisters. So we needed to avoid complete overlap of our variables, that kind of thing. we saw that family Lactobacillus was associated with this interaction. So those who don't express lactase but consume a lot of lactose had a lot of Lactobacillus. So what does this mean? Now it's the same frustration sort of with 16S data. There's this weird correlation. It's only at the family level. How do we interpret this? Well, Danielle and I, who worked on this project together, I should have said earlier, Elizabeth Chin was in Danielle LeMay's lab at the time, feel like this could be due to bacterial cross-feeding. So because this wasn't an intervention, we're capturing the amount of lactose that people consume habitually. And if they are voluntarily consuming lactose, we can make the big assumption, perhaps, if we're just generating a hypothesis, that they have decreased discomfort with lactose consumption. And the genera that are most predominant, most common in our data set within that family are Blautia, Roseburia, and Coprococcus. So the great and interesting thing about Blautia is that many species within this genus can convert hydrogen and carbon dioxide to acetate via acetogenesis pathway. And this might compete with methanogens in the colon, which also use hydrogen and carbon dioxide for production of methane through the Wolff cycle, which is something you would want to avoid when consuming lactose. Also, Roseburia can produce, among other things, hydrogen and carbon dioxide. So we were kind of hypothesizing that maybe these work together in a very closely knit feedback loop, because also Roseburia and Coprococcus can metabolize acetate to butyrate. And so these individuals perhaps could consume lactose comfortably and get a fiber-like benefit of producing short-chain fatty acids rather than uncomfortable gas. So I also just want to make a plug. Stay tuned, because we are investigating those hypotheses. So hopefully really exciting results to come. And Dr. Lorene Coates in my lab has also been working on how fiber type consumption might affect the milk microbiome. And her work was primarily in bovine cows through the USDA Dairy Grand Challenge, but it's also pretty exciting. So stay tuned. And with that, I just want to acknowledge everyone who worked on the phenotyping study, my amazing collaborator, Danielle LeMay, who's pictured here, everyone in my lab, and also these creatures who I take inspiration from, because they utilize the stool from other organisms, organize it, and make the world a more beautiful place. And with that, I will either move on to questions or move on to Danielle's talk.
Great, thank you so much, Dr. Cable. We are going to go directly to Danielle, and then we'll take questions at the end. So I am pleased to introduce Dr. Danielle LeMay, and Dr. LeMay is a research scientist at the Western HNRC. She's also an associate adjunct professor in the Department of Nutrition at the UC Davis. She's also the nutrition cluster lead at the USDA-NSF AI Institute for Next Gen Food Systems. And her lab uses lots of interesting techniques like bioinformatics. She applies omics technologies and machine learning AI in her work. So we'll hear about some of that today. So we are looking forward to hearing from you, Dr. LeMay.
Great. Can you confirm that you see my slide?
Yes, I confirm.
Thank you. Thank you. Thank you for that introduction. I'm happy to talk with you all today about eating for trillions, which of course I mean the trillions of gut bacteria. And the relationships between diet and gut microbiome and gastrointestinal health. So these are my conflicts of interest, or disclosures rather. Although to be clear, I'm personally only paid by the US government as a government employee. and don't receive further compensation from these other activities. Okay, so I like to think that everything we need to know about nutrition science, we can learn from looking at mother's milk. This is the only, the only nutrition that a baby is receiving for those first 6 months, sometimes beyond. So what does mother's milk teach us about the food that our gut microbes need? Well, one of the most abundant components in human milk are in fact not digestible and absorbable by the baby, but are there for the baby's gut microbes. And I'm talking about human milk oligosaccharides. So after lactose, lipids, HMOs are like the next most abundant component. So these human milk oligosaccharides, I've just got a cartoon of them here where, like, you can see the monosaccharides are just these shapes, and then the dotted lines are the linkages between them. And so this is just a cartoon of a canonical structure, but there's actually 200 different HMO structures. And the thing I want you to remember here is just that, look, there's all these different linkages, and then to get at those monosaccharides, the microbes need to have all different types of enzymes to break that apart and use the monosaccharides for energy. In addition to the HMOs, there's also carbohydrate structures that attach to the peptide, the proteins. So the N-glycans and O-glycans are decorations, carbohydrate decorations of proteins. And there's even glycolipids, which are conjugates of carbohydrates and lipids. So the take-home message here is that there's a lot of carbohydrate complexity in human milk. And that carbohydrate complexity is there for feeding gut microbes. And so that tells us that, our diet probably needs a lot of complex carbohydrates to be feeding our microbes. So what I'm going to talk about for the rest of this talk are three prongs of that. And first, diet. How do we capture dietary components that are influencing the microbes? Because it's not as simple as the summary variables from ASA 24 or a food frequency questionnaire. And then second, how do we measure how gut microbes are responding to those components? And then finally, how does all of this impact our gastrointestinal health? So beginning with diet, how do we study diet microbe relationships in humans? Well, today, we are often using something like a food frequency questionnaire, which is the example I show on the screen. And so what our dietary data actually looks like is things like, this person consumes sweetened cereals, 3 to 4 times a week. Well, it's very hard to reduce that down to what are the carbon sources in the diet. So the food frequency questionnaires and the 24-hour dietary recalls, we are obtaining those dietary components or estimating those dietary components from food composition tables. The USDA Food Data Central is an excellent database that's used worldwide, but it does only have 69, nutrients for each food. But we know that there's thousands and thousands of molecules in food, and those are not captured today in our food composition databases. And so we refer that to sort of the dark matter of food. There are advances in analytical chemistry that are quantifying more and more of that dark matter. So our lab takes 3 approaches to improving dietary data in a way that we can capture food components that are important to gut microbes. The first is improving the dietary intake instruments themselves. Second is collaborating with analytical chemists, I'm not a chemist, to improve the resolution of diet, to capture those thousands of molecules. And then the third is using new methods that can identify among, thousands of things what's important. So in the area of new dietary instruments and using food photos. One of the first issues was there was really not any kind of benchmark for assessing how well do we capture diet from a food photo. And so we worked on this clinical trial in which we recruited people to capture their diet both with photos and with traditional food records, and spent a lot of time QCing these and linking each photo with each line of a dietary record. And we have released this benchmark now on USDA Ag Data Commons. We've applied the benchmark to some existing algorithms and working with some computer vision experts to improve those algorithms. In the area of chemistry, we've been collaborating for some time with Carlito Labria on the Glycan Encyclopedia of Food. The first version is already released, and that captures the abundances of 14 monosaccharides across 800 different foods. The second version will be released soon. There's a manuscript already, and that one captures monosaccharides, both free and total, as well as glycosidic linkages and some information about at the polysaccharide level. So why does all that chemistry matter? We can map those foods now to our own dietary data in our cohorts. So Mary already introduced the USDA nutritional phenotyping study. And from that study, we took the dietary data, mapped it to the glycopedia, and we were able to deduce, what were the most common food sources that were associated with particular monosaccharides. And so, for example, avocado appeared to be the most important source of GAL-A and rhamnose. Coffee was actually the most common source of mannose and rhamnose. And then a more expected relationship, whole wheat bread was a source of arabinose and xylose. And these are just some examples I'm pulling out. We published the paper if you want to dig in further there. Now, the next issue is, so Dr. Cable did already explain the food tree, which is great because then I don't have to explain that. But we're taking that now one step further. So with traditional dietary analysis, you're looking at summary variables, for example, all grains, all meats, all vegetables, and so forth. With the food tree, you're able to actually specify those relationships, but there is the issue, as there is with microbiome science, of, well, okay, what taxonomic level do I now analyze my data at? So Dr. Oliver in our lab has created a new software package to do some feature reduction with a hierarchical feature engineering step that allows us to do that feature reduction with information that we already know from that taxonomy. So it's a taxonomy aware method. And here is just a toy example of how, how we can use that. The example is, let's build a model from the food tree to find what are the contributions to fiber intake from a healthy adult cohort. So you can see that the biggest predictor was a level 2 node for non-citrus fruits. And the other two, there's a level 1 node for beans, peas, and legumes, and a level 1 node for vegetables. Those two you would have found with a traditional analysis, but then there's also a level 2 uncooked cereals. And so that's a good example, like the cereals, the uncooked cereals being more important than the overall grain category is something you wouldn't find in a traditional analysis, but it's certainly makes more sense biologically. Okay, we're going to move on to gut microbes. How do we study their response? This is kind of a busy picture, but I just want to make the point that we do know a little bit about how certain foods will cause a bloom of certain bacteria to grow, and depending on which microbes are growing, they produce different byproducts. So this picture makes it sound like we've sorted it all out. We haven't, but the take-home message here is there's a lot of complexity, and we need to drill in further to completely, map this out for all microbes. So how do we study diet microbes relationships in humans? It's pretty tough, in that, well, we can record their diet, but we have limited points of observability, and usually we're making use of the stool, which as an approximation for the microbes that make it to the distal gut. And we can use, DNA from the stool to identify which bacteria are there, RNA to identify, which bacteria are active, and we can also measure microbial metabolites. So one thing my lab has really focused on has been improving bioinformatics methods to capture these microbial responses. So like in 2016, we put out the first metatranscriptomics software that leveraged a public metagenomics server. 2018 released software that just runs independently on your own hardware. In 2020, we released new methods for functional annotation of human fecal metagenomes. And then most recently, we now have this new methods for machine learning for microbiome data. So I just wanted to give one example of where metatranscriptomics was super helpful in our research. And this was for rhesus macaques with idiopathic chronic diarrhea, meaning we don't know what causes this chronic diarrhea. We did not make any monkeys sick. These are animals that spontaneously become sick, and then we match some healthy controls. Now, interestingly, the healthy and the sick monkeys actually consume the same diet. And many of the same microbes are present in the healthy and sick animals. But what the microbes are doing in those animals were different. So with the metatranscriptomics data, we could see that the Campylobacter that was in the sick animals was expressing an adherence protein, CADF, that enabled it to adhere to fucosylated mucins in the GI tract. Another mechanism was one of the most abundant microbes, the Bacteroides fragilis, was expressing a fucosidase that enabled the cleavage of that fucose from the fucosylated mucins, and that liberated the fucose, and then other not so great bacteria were able to take up that fucose. So we call that cross-feeding, when one bacteria is breaking up a complex carbohydrate into something smaller that another bacteria can then feed off of. So these were two different mechanisms that may be driving this chronic diarrhea that was really only possible to see with metatranscriptomics. Okay, so another area of concern for gut microbes has been antimicrobial resistance, and it's really a global concern at this point. And so we measured antimicrobial resistance gene abundance across our healthy cohort of adults in the USDA nutritional phenotyping study. This was, here we had shotgun metagenomes from 290 people. Here they're ordered in abundance of antimicrobial resistance genes. And you can see that even in a normal healthy cohort, there's a lot of variance. And there are people with much higher, some people have much higher antimicrobial resistance genes than others. So we did some machine learning to try to figure out what were the predictors, dietary predictors of antimicrobial resistance. And we found that the strongest dietary predictors of being in the lowest quartile of AMR, meaning having the least amount of antimicrobial resistance, was an increase in dietary diversity, or soluble fiber, or total fiber. So it's nice to, every now and then, to have that satisfaction that diet does matter. Going back to our monosaccharide story, where we had used, the new Davis Food Glycopedia to see which foods were more abundant in our healthy cohort, we're able to extend that to the microbiomes of these people in that cohort And so we could see, for example, people who were consuming high arabinose also had higher levels of ruminant clostridium, and the flip side, like lower levels of Blautia. And then people who were consuming higher GAL-A had higher levels of lactospira. This is fitting with what Dr. Cable showed you with the relationship between fruit and lactospira. But the take-home message here is that, even looking at the level of monosaccharides, we could see that there were relationships with particular gut microbes. And so it may be possible in the future to take this information and tailor diets to help modify the microbiome. Okay, so finally, how does our diet affect gastrointestinal health? Well, one reason we think diet can impact GI inflammation is all the studies that have come before us in mice, where mice fed a diet that's depleted of dietary fiber, have impaired gut bacteria. They have, more leakage of bacterial components into the bloodstream. They have chronic inflammation. They are more prone to infection with enteric pathogens. But this has all been shown in mice. And what happens in humans? Well, when we are studying healthy humans, we can't just stick a cannula in them. We can't take biopsies. So we have to rely on blood, self-reported GI symptoms, what we can learn from the stool, and markers of inflammation. So one really easy place to start is measuring fecal consistency. The fun thing of matching the stool to to the Bristol stool scale. And so the Bristol stool scale showed here is a 7-point scale, depending on the hardness all the way to normal to softness of the stool, with type 4 being considered the ideal. So we measured the Bristol stool scale form score in our cohort of healthy adults. We also asked a subset of those adults to self-report their own Bristol stool score. And this is the first time this has been done. So it was really interesting to see that, most people thought their stool was ideal. So people think their poop is normal consistency, but often it's not. Like the technician who scored everybody's stool as it came in found that many people had like hard or soft stool. So this was just an interesting finding on the discrepancy of self-reports, but also the fact that a lot of people are having hard or soft stool. And what are the dietary factors that contribute to having a not normal stool consistency? Well, it turns out there weren't really any that had super strong effects on their own, but certainly we did with some machine learning, we found that it was really a constellation of dietary and other factors, especially stress hormones, that predicted stool consistency. So as one would expect, eating more vegetables, having a better unsaturated to saturated fat ratio, and total dietary quality as measured by the Healthy Eating Index score, those all were strong contributors to having a normal stool. And then finally, how do we measure GI inflammation? As I mentioned before, we can't take biopsies, but we can assay with ELISAs proteins and other markers that show up that appear in the stool that are derived from immune cells that are in the gut. So on your left, there's this picture, cartoon of a colon, and all the cells with the green dots are your intestinal epithelial cells. And you can see that other immune cells do migrate into the intestinal area. The fecal markers that we use are calprotectin. Calprotectin is high in neutrophils. It's also kind of lowly expressed by intestinal epithelial cells, so it's somewhat of a non-specific marker, but it is used in clinical study. It's used as a clinical diagnostic tool, and calprotectin is really resistant to digestion, and just it's stable for longer, and so it's a good marker in that respect. Myeloperoxidase is high in neutrophils, and neopterin is high in macrophages. So our hypothesis was that dietary fiber would be associated with GI inflammation, and we did find that even in healthy adults, as you increase the fiber in grams per day, there was a lowering of neopterin. So I guess the rules apply still as we expect, but of course, because these are human participants, the data is still there's still a lot of variance in the data. We also found that high diet quality in the form, using the healthy eating index was also associated with lower GI inflammation. And then one last point is, beyond the gut, there's more distal sites that are of interest, like bone or brain, and what's happening in our gut may well impact those distal sites. So this was a machine learning project that was conducted by Dr. Chin, and she basically took the data from our nutritional phenotyping cohort to build models and explain what are the contributors to bone mineral density. And of course, she found the expected contributors, like our ethnicity is a strong component, age, sex, your body mass index. Those are all strong predictors. But we wanted to drill further into other features that are not that are modifiable. So and the feature that really popped up was the stool pH as an important predictor, and so this makes sense in that lower stool pH is indicative of more fermentation of fibers, and also the lower stool pH would enable more absorption of calcium, and that's the bone connection. Okay, so I just threw a lot of stuff at you today, so I'm just going to boil it down to a few things to remember. One is we're developing new methods, food photos, analytical chemistry, data science, to identify dietary components that are important to microbes. We did find that higher fiber diets are associated with gut microbiomes that harbor less antimicrobial resistance. Particular monosaccharides in the diet are associated with specific food sources and abundance of particular microbes, suggesting that it may be possible to alter gut microbiomes with tailored diets. And then finally, higher fiber diets are associated with lower gut inflammation, even in healthy adults, and may impact distal sites like bone. So thank you for listening. Science is a team sport. I particularly want to call out the work that a lot of the work I featured today was work by some brilliant postdocs in my lab, Dr. Elizabeth Chin, Dr. Andrew Oliver, and Dr. Jules Lark, and graduate student Yasmin Bouzid. And none of this would be possible without the huge team that produced the USDA nutritional phenotyping study data. And Dr. Charles Stevenson was the PI of that study, and Dr. Mary Cable, who's our microbiologist, who I work with closely. So thank you. I look forward to your questions.
Great, thank you, Danielle. If you want to just stay on webcam, and we can have everybody else come back as well. Great. And Dr. Stevenson, I think you're about to come back. Okay, perfect. Great, welcome back, everybody. Thank you so much for that whirlwind of information. Maybe we could start with Dr. Stevenson talking a little bit about how the research priorities are determined at the centers. Maybe you could talk a little bit about timing of those cycles, considering a lot of these studies, especially for feeding studies, are going to take 4 or 5 years from start to finish. How does that work?
That's right. Particularly our clinical studies take several years. So we have 5-year cycles for our research program. So all 6 of the Human Nutrition Centers are part of National Program 107. And so we have a national program leader who organizes input every 5 years from stakeholders and comes up with a plan. So our current plan is actually ending this year, and the new NP 107 plan 2024 through 2029 is up on the web. If you Google human nutrition, USDA Human Nutrition Program 107, you'll find it. And they're broad, basically 5 broad components that change a bit over time, but one new emphasis is on a bit more on food systems, so how ag systems affect dietary quality. Of course, the Beltzfeld group is very interested in food composition and maintaining the nutrient databases. As I mentioned before, scientific basis of dietary recommendations. So a lot of our work in all the centers falls into that area. So if Danielle and Mary's data that higher fiber has results in a lower antimicrobial resistance burden, that could well be shown in the future to be a direct health benefit. So that's one example. Prevention of diet-related chronic disease is another category. And then life stage nutrition. 3 of our centers are focused on infancy and childhood in Arkansas, sort of childhood adolescence in Houston at Baylor, and then more on the other end of the lifespan, geriatrics, older adults in Tufts. So those are broad categories, but then really each scientist in the 6 centers or groups of scientists, like our Immunity and Disease Prevention Research Unit, there are 5 of us. We just wrote a proposal for 5 years of funding, essentially 2024 through 2029, and every scientist in the Human Nutrition Program has been doing that this year. Those proposals have gone off to peer review. There's an external peer review, mostly academic scientists. And the review period, at least for our group of programs, that's all being done this year. We're expecting reviews back in November, hopefully. So we get external peer review on the specific research our scientists propose, respond to that, and hopefully with all reviews, there's a little anxiety around that, response to reviewer comments, certainly at a minimum. And then in early 2024, our new 5-year plan will begin with new specific projects in those that are somehow covered by those 5 major topic areas I mentioned.
All right, great. It sounds like a substantial amount of work. So I understand why we had to move the schedule for these webinars until after I think that submission was complete. Right. So fair enough. Okay, so maybe a question for Mary, which is about the surprising relationship between French fries and gut microbes. And it's just asking for your thoughts on whether that might be due to the way commercial fries are prepared. So I don't really know if they're cooked, then frozen, then cooked again. So there's some resistant starch implications there.
Yeah, and we, that was our thinking as well when we saw that relationship, because some of the other foods in that cluster were cooked corn and other ways that you would generate resistant starch type 3 from heating and cooling resistant starch type 2. So actually we are testing that exact hypothesis. Dave in the lab, because it turns out to be really expensive to buy resistant starch type 3. So Dave has been cooking and cooling potatoes and and isolating the resistant starch type 3 out of those. It was a huge task, and fermented those in our continuous flow fermenters. So we tested that hypothesis, and I don't know the answer yet, so stay tuned.
Okay, we'll have a whole new way we should be cooking our potatoes that takes forever after this.
Maybe
Yeah.
not.
Okay. So Danielle, a question for you about your efforts to improve dietary assessment with the photo capture of foods. So can you talk a little bit more about how you're linking the photo to the energy and nutrients in the food? That seems like a big challenge that might still require human input for all of time, or input from the actual consumer still. So maybe talk a little bit about that.
Right. So first of all, there are apps on the market today that do this, and I suspect the way that they've been doing it is with labeled data sets and neural nets. However, there's been no there hasn't been a benchmark for, well, how good is it? You know, there's this thing on you can buy an app and use an app, but there's no like science behind it to know how good it is. So the first step that our lab did was create the benchmark. And then I didn't actually present any results today on how we're approaching this problem of connecting foods to their nutrients, but we are leveraging with our collaborators what the latest in generative AI. There are now general purpose image to text models. It's fascinating, you know, that these things work as well as they do, and so, you know, we're making gains in that area. I also want to say that one of the things that we're attempting to do with these models is to predict the actual ingredient, and from there mapping to a special interest database. So it could be, so in addition to the nutrients that are provided by the USDA, there's, you know, like the food glycopedia, there's food DB, there's other special interest databases that are popping up as analytical chemists are filling them with, you know, all the molecules in food. So in predicting at the ingredient level, we're able to then map to those.
Okay, all right, so possible, still some work to do on that.
Yes, work in progress.
Yep, okay. Okay, so still for you, Danielle, there's an FNB meeting that's actually still going on right now that's about AI basically in food and nutrition research. And one key point that was made there is, at least what I took away, is maybe now is not the time to get answers using AI, but hypothesis generation, sure. But a key point was now is the time to establish systems and structures for data collection to make sure that it can be leveraged to use AI to answer questions in the future. But how do you see the opportunities and challenges for AI in this space? Right.
So there are immediate opportunities and challenges, and sort of some long-term ones. So first, I'll start with opportunities because that's the fun part. You know, AI can help design functional foods. There are startups that have spun off that are doing just that. So you can build more foods. AI can also help make predictions about how to reroute, you know, commodities or waste streams in ways that help get more out of our food production, and then more for a better food distribution. And then, you know, from the nutrition side, like AI can help us do better dietary assessment and dietary recommendation for people. So dietary, you know, recommendation systems that will tell you, you know, what you should eat today to improve your nutrition. But there are a lot of challenges. So like you mentioned, you know, the data that you're collecting is really important. Whatever data you're using to train the model will be limited by the quality of that data. So that is one concern is how to collect enough labeled data. Generative AI is using lots of data that's really being collected from all over the place. So I completely agree with the idea that we should only use it at this point as a brainstorming method, not a method to tell us truth, because it will, it will hallucinate, like it will lie. If I ask, ChatGPT some things about diet and gastrointestinal health, it makes some things up, and if you ask it for a citation, it'll be, oh, I'm sorry, I don't have a citation for that. So yeah, at this point, I would say brainstorming only. But we as nutrition scientists often think about clinical trials and all that as like who's incurring some risk and who's benefiting. And I think a big challenge in AI is going to be like, what are the ethics surrounding, data collection? Who's incurring the risk of that data collection? Who's benefiting from the models? And we at USDA, we want to, of course, democratize benefits as much as possible. So those are all a lot of challenges. Like when you think of the recommendation system, who's going to pay for the first ones? Well, it would be people with those just extra money to spend. So, but if you only tailor to their needs, then how do you meet the needs of people who don't have money to spend on an app? So those are the kinds of things that we wrestle with and the challenges.
Yeah, and that brings up another point in the meeting, which was about ensuring the model includes data from all different types of people, different socioeconomic status. Otherwise, it's just giving you an answer for a certain type of person. Okay, thank you. And a question for Mary then, which is focused on lactose intolerance. So is there any other way people really deal with lactose intolerance besides abstinence? And then do you see, in terms of treatment of the condition, any therapeutic applications of your findings or your hypotheses so far, depending on where they go?
This is a tough question. It's one of those things like I sort of want to politician it, so you can't hold me to anything. But I'll just say what I really think based on the literature, which is if you want to be able to eat lactose, you'll have to force yourself through the discomfort until you get those bugs that are going to be more acetogenic and less methanogenic. And probably the way to do that would be like a step increase. So gradually eat more and more lactose. And I think it's over a really long period of time, because like most of the intervention studies didn't capture it. They just increased, or they just captured the increase in lactic acid bacteria, which is what a lot of studies are capturing. So that's what I think. I'm not sure it would really be a therapeutic application at this point. I mean, maybe someday we could give acetogens as probiotics, because I feel like they're not a primary focus at the moment. We're more focused on lactic acid bacteria. But people also take lactaid. That, of course, won't give you like a fiber type benefit from lactose, but you can, eat ice cream in public. So anyway, that's my 2 cents.
Okay, thank you. Thank you for speculating there. And then Danielle, to the continuing on the dairy theme, but the question is, to what extent do bovine milk oligosaccharides exist in the commercial milk supply, if you know that?
In very small quantities, actually. So to put this into perspective, a supplementation trial where we fed people bovine milk oligosaccharides, to get enough grams of bovine milk oligosaccharides for a supplement, we needed like 14 liters of milk. So drinking milk for the bovine milk oligosaccharides, there aren't going to be a lot of them in it. That doesn't mean there's no benefit, because people drink a lot of milk over a lifetime, but it's a lot smaller than you might expect.
Okay, interesting. So I'll stick with you, Danielle, and shifting a little bit to a hot topic of today, ultra-processed foods. Do you know of any research looking at this category of foods, also called Novel-4, and the impact on gut microbiome, which is obviously going to be looking at all the components of those foods as they are currently defined?
Yeah, so the NOVA classification scheme is really imperfect, as you probably know, and because you've got things that are because even yogurt is considered a processed food, right? But it's highly beneficial, and as there are more functional foods coming on the market, there will be some ultra processed foods that are designed to be healthy, and then there's other ultra processed foods that are just designed to be tasty, and so the NOVA system doesn't really capture any of those differences. I will say there's a lot of work with cell cultures that show us that things like the emulsifiers and gums and additives and all that are not good for gut barrier function and can cause inflammation. If you think about the mucin that lines our gut, throwing in a lot of emulsifiers to break up that mucin, probably not a good idea. But again, it's the amount of dose, and I haven't seen this studied in humans, and that's what we do need this to study that in humans, especially as more and more processed foods are coming onto the market. We need to have a better understanding of which components are okay, what doses. Yeah.
Right. Yeah, and I think just to your point about better dietary assessment methodology, we don't know a lot about dose in the current food supply, which is always changing. Yeah, so okay, so Mary, a question for you about low FODMAP diets, which are recommended by GI physicians oftentimes to treat IBS. So that's going to restrict all those oligosaccharides that are included in the FODMAPs. Do you have any thoughts about that diet? I don't know if you know a lot about it, but what the impact might be for health. Any feelings?
So this is a complicated question. I feel like elimination diets in general are somewhat risky because you're eliminating nutrients that you might be getting and you're eliminating diversity. However, I will say that like the study that Danielle showed where we were looking at ulcerative colitis model in monkeys, where their microbiome has sort of turned against them, so that even if they eat a regular diet, their microbe is going to make that a weapon. That's how I think of it. And I think that's what physicians are going for with that elimination diet. They're trying to reset so you don't, so that you can try and change the transcriptomics, like the genes that your microbiome is expressing. And I think that has a long way to go still, understanding that and finding a way to fix it. And I think FODMAP is just, physicians are doing the best they can with what they have for people right now who are suffering.
Okay, thank you. I'm sorry we're giving you all these hard speculative questions, but you're doing great. Okay, so maybe one more question for Danielle, which is about antimicrobial resistance. So is there a way to measure that, your individual human degree of antimicrobial resistance, and can you reduce that?
Is there a way to measure that in for yourself? Is that what you're asking?
Yeah.
So research-wise, we can certainly measure it by, measuring the DNA from your fecal samples. There's not a commercial app. I'm not aware of a commercial provider that measures that for individual people. But the other thing is, we're all going to be exposed to antibiotics at some time in our life. So I think that the overall message here was, consume a diverse diet that's high in fiber as more of a prevention mechanism. Yeah.
Okay. And the complement question is whether or not a higher fiber diet in food animals might decrease antimicrobial resistance.
Yeah.
In the product that we eat, and then there are benefits for the food.
Right. Great question. So Actually, how we came to do this human study was we were collaborating with David Mills's lab looking at antimicrobial resistance in dairy cows. And we found that the colostrum being fed to the cows was really high in antimicrobial resistant genes already. So the baby cows were sort of seeded with a lot of antimicrobial resistant genes. And then as the baby cows were weaned and put on the higher fiber diet, that crushed, over time the antimicrobial resistance. So seeing the impact of fiber there made me question, okay, well, in humans, what do we see? And so from the dairy animals, the first thing that comes to mind is they probably need to change some of the husbandry practices. So because the colostrum is sort of a pooled colostrum, but it does. Thinking about in animal production, that initial phase of how animals are reared and whether the milk is a source of antimicrobial resistance or not, and is a pretty important thing to consider. And then what's the weaning diet? What we haven't you looked at, so it's not our mission, but I mean, we as scientists need to do more about looking at how changing the fiber content of the feed, how does that impact antimicrobial resistance in animals? So I think that the dietary component is certainly meaningful even in animal production systems.
Mm-hmm. Great, lots of applications then. All right, I think we will close on that for today, and we hope that everyone will join us again on October 24th. So we'll have the second in our Western HNRC series that day, and that will be focused on diet, stress, and immune function. So please go ahead and register. And we just want to give a huge thank you to our excellent speakers, and really appreciate you sharing your work with us and enlightening us as to what's going on at Western HNRC. And for those interested in finding more about IAFNS and our projects, please go ahead and visit our website and also look at our upcoming events and register for those. So thanks so much, everyone, and we hope you have a great rest of your day.